Hello and welcome to the mind map about enzymes for Year 11 biology. Okay, we're going to zoom in. Enzymes are organic catalysts. Organic means that it has a complex carbon backbone. In fact, enzymes are proteins made of a chain of amino acids. Being a catalyst means it speeds up the rate of reaction. It does this by lowering the activation energy required to initiate the reaction. Over here at the top of the left-hand side, we've got a diagram that shows you the effect of an enzyme on a reaction. In the reddy pink diagram, we've got an input of energy required called the activation energy for the reaction to take place. In the green, we've got the effect of what the enzyme does. It lowers the activation energy and therefore it allows the reaction to take place quicker. Okay, so the substrate is the name of the reactants in the reaction. The products are what are made in the reaction. Specificity means that the enzyme can only work upon specific substrates. The active site is a pocket in the protein where the substrate fits. The enzyme is reusable because it's not used up by the reaction, it just stays the same. We say that it participates in but is not used by the reaction. There are generally two types of reaction. There's anabolic, which is endergonic. Anabolic means that it builds up molecules, and endergonic means it uses up ATP. Then we have catabolic, which are generally exergonic. Catabolic means that it breaks down molecules into simpler ones, and in doing so, it releases energy. And in the cell, we store that energy as ATP. Some enzymes can catalyze both the forward and the reverse reaction, and we'll talk about this more when we're talking about product concentration. Next, we're going to talk about how enzymes actually work, and the old theory was called the lock and key theory. The substrate approaches the active site. Hydrogen bonds form between the substrate and the active site. As the substrate enters the active site, it then forms a necessary step called the enzyme substrate complex, and then products are released from the active site. The latest theory is called the induced fit theory. This states that the active site is not the perfect shape for the substrate, but twists and contorts to allow the substrate to fit into the active site. Then the rest is the same. Hydrogen bonds form between the active site and the substrate. The substrate enters the active site. We have the formation of the enzyme substrate complex, and then the products leave the active site. Then we come to the factors that affect enzyme activity. The first is temperature. As temperature increases, the kinetic energy of the molecules increase. Therefore, there's more collisions and the enzyme activity increases. The optimum temperature means that there is the optimum number of collisions between the substrate and the active site, and therefore at the optimum temperature, we get the highest activity. After the optimum temperature, the heat starts to break the hydrogen bonds between the amino acids, and the enzyme unravels, losing its active site. It can no longer form the enzyme substrate complex, and therefore if it can't form the enzyme substrate complex, you don't get any product, and we call this denaturation. At the top right-hand side, we have a wonderful diagram about the effect of temperature on enzyme activity. And as you can see, after 50 degrees Celsius, the enzyme starts to denature and the rate of reaction decreases. And then we have pH, which is acidity. Some of the amino acids are charged at different pHs. As the pH changes, the charges on the amino acids also change. And these changes can stop hydrogen bonds being formed between the different amino acids. And again, the enzyme unravels and you lose the active site. If you lose the active site, you can't form enzyme substrate complex. And if you can't form enzyme substrate complex, you can't get any product. And we call that denaturation. For both temperature and pH, denaturation is irreversible. Once it's denatured, the enzyme is useless. Then we get down to what we call enzyme kinetics. First of all, we have the effect of enzyme concentration. And as the enzyme concentration increases, the rate of reaction increases. As more active site becomes available, more enzyme substrate complex can be made and therefore more product can be made. Next is substrate concentration. And as the substrate concentration increases, the collisions increase and therefore the rate of reaction also increases. This happens until the active site is saturated and can't work any faster. And this is where the level of activity levels off. And we call this the saturation point. Next, we have product concentration. As the product concentration increases, the product can inhibit the reaction taking place at all. We call this negative feedback. In fact, as the product builds up, it can drive the reaction in the opposite direction for those reversible enzymes. And we've got a little diagram here about the effect of the buildup of a particular product feeding back to an original enzyme to stop it from working. And that is, in fact, called negative feedback. Some enzymes require cofactors and coenzymes to modify the shape of their active site to accommodate the substrate. Cofactors are inorganic. For instance, magnesium is needed by the enzymes in glycolysis. And coenzymes are organic factors, normally vitamins, and for instance, we've got B6, which is used for the production of red blood cells. Inhibitors block the active site. In fact, we should say that inhibitors change the active site. Competitive inhibition is where a molecule or a substance competes with the substrate for the same active site. And an example of this is magnesium and calcium during muscle contraction. This is why magnesium works as a muscle relaxant, because it competes with calcium to stop the muscles contracting. Non-competitive inhibition allows for a molecule to bind to another site, changing the active site. And the example here is cyanide reacts with cytochrome C in the electron transport chain to stop cellular respiration. And next we get to ATP. ATP stands for adenosine triphosphate. It's made by the enzyme ATP synthase, which is abbreviated to ATPase. ATPase is found on the internal membranes of mitochondria and chloroplasts. In mitochondria, the internal membrane is called the cristae, and in chloroplasts, the internal membrane is called the thylakoids. The substrates to make ATP are adenosine diphosphate, or ADP, and an inorganic phosphate known as PI. It also requires the cofactor of a magnesium ion, here we are, Mg2+, and requires the flow of hydrogen ions through the ATPase. Of note here, we should also mention NADH and FADH2, which are produced in cellular respiration and donate electrons that allow the hydrogen ions to flow through the ATPase. Okay, thank you for listening. Don't forget to like and subscribe, and I'll see you next time. Goodbye.